this picture yesterday? Yes. All right, what did we label? That thing.
Nucleolus. Oh yeah, that was the last thing we did.
And did we label this thing?
Chromatin. Chromosomes. Chromatin. Synthesis.
And those things?
Centrioles.
And that? The nucleus, which is held together by the nuclear membrane or the nuclear envelope. So nuclear membrane or the nuclear envelope. So all that's familiar. We all know the players in the cast. Oh, and of course, this thing is the cell membrane, which I think it was Cameron who told us that was a phospholipid bilayer. See, it might all blend together, but it's just because I think you're in both 6th and 7th hour. Did we look at a picture like this?
No.
Okay. So in order to fit all 6 billion nucleotides of DNA into our cells, you know that A, T, C, G nonsense? To get all 6 billion to fit, we have to package it pretty efficiently. So your body is going to take it, which would have been this in the form of DNA, and it's going to wrap it around some proteins that you see up here. That protein is called a histone. When it gets wrapped around histones, that's when it becomes chromatin. 5th time today I've gotten hiccups. Those histones are going to form a complex of like 8 histones called nucleosomes. And when it does that, it looks like a little flower. So that's what happens right here. Those nucleosomes are going to wrap up even more tightly. And when they wrap up even more tightly, they form a chromosome, which is what this whole big picture is. One big chromosome. And just to kind of make the whole picture clear, here's what I'm describing to you. You have all worn a piece of clothing we call a sweater before, right?
Yes.
Excellent. I'm glad to know we all have something in common. And you understand that sweaters are made of a single thread that gets woven together, right? And if you pull on that thread, you unravel the whole sweater. Agreed? That's all I've done here, is I've taken a chromosome, and I'm about to use the laser pointer that makes noise. I did this without warning previous hours, and they were like, I want you to be warned. Laser pointer with noise. Yes.
I'm confused about histones.
It's a small protein that the chromosome wraps around. So it's just a little ball of protein.
Okay.
Consider it like a spool. Do you know what a spool is?
Oh, I don't know. Like
A thread?
it wraps around it like it's.
It's Wednesday.
So the chromosomes are made out of the protein.
So all a chromosome is DNA wrapped around a whole bunch of proteins. So what I've done here is shown you a chromosome here that I've just pulled a thread out of, and I kept pulling it, and if I pulled and pulled and pulled and pulled, the whole chromosome would unravel. Anybody have a sweater they want to donate? No, you don't really want me to unravel your sweater.
Can I unravel this one?
You can't unravel that one,
Yeah.
because it's not a sweater.
Yeah, it is.
No, AJ. It's a
Like, have you ever seen like a Christmas sweater?
yeah.
Preferably an ugly one. The example is made better if it's an ugly sweater.
You have it?
There's no such thing as an ugly sweater.
Oh no, I ran in a hideous holiday sweater run.
Oh, yeah, but the Christmas ones are horrendous.
Yeah, there were blinking lights in my sweater.
Maybe horrendously flashy, but they're not ugly.
It was really horrible. Oh. Chromosome. If I pulled one thread out and kept pulling, it's going to take up a lot more space than the original chromosome took up. Now the cool thing about our chromosomes is actually it's composed of two different legs that I will outline in different colors here. There's one, and here's the other. Those halves of the chromosomes are called sister chromatids. Those sister chromatids contain identical information. So imagine I made a copy of something and then stapled that copy to the original. That's what this is. At the center, instead of using a staple, we use this yellow thing, or orange, whatever it appears to you guys. On my iPad, it looks yellow. Up there, it looks orange. That thing is called a centromere. And its job is to staple two identical sister chromatids together to keep them organized. When I say they're identical, I mean, ooh, that color, that the one that I am about to draw diagonal slashes all over contains exactly the same genetic information as the one that I'm about to draw stars on. Those two are identical to each other. So if this happens to be the chromosome that contains the gene that you guys have for whether or not you can smell asparagus in your pee, I had to pick an interesting one to make sure you're paying attention. If it's located right here on this sister chromatid, that stretch of that DNA codes for smelling asparagus in your pee, it's located right here on this chromatid as well. So this is the copy that you would have gotten from mom, and this is the copy that you would have gotten from dad. Those two copies together indicate whether or not you can in fact smell asparagus in your pee. How many of you know what I'm talking about?
I don't usually smell my pee, so I don't.
Have you eaten asparagus?
No.
Eat asparagus. Come back and talk to me. You can't help it.
What, eat asparagus?
No, smelling it in your pee.
Smells bad.
It smells like asparagus.
It smells like pee. Pee doesn't
It smells like asparagus.
All right.
I don't really like your tone.
Perhaps asparagus is a bad example for you. Instead, let's say this is the gene that turns your pee pink after you eat beets.
What?
Real?
Does that happen?
Is it real?
You have to eat a lot.
It's so
I'm eating a
weird.
ton of beets now.
Well, the more beets you eat, the more colorful it gets, so be careful. It turns out magenta if you eat a lot of beets. But not the yellow beets. Don't go to the store purchasing the golden beets, assuming that golden beets are going to turn your pee magenta. It's got to be the red beets.
So half is from your mom and half is from your dad?
One side is, yeah.
So how are they the exact same then?
They contain the same genes.
Oh.
And you'll see why that's important in a second. In some people, something weird happens, which is called genetic recombination, and then it's going to be like the same one on each leg for mom and mom on the other side. But that's like 2 chapters away. So that's what we'll get there. I know you've wanted to learn this much about urine today. Welcome. You got to choose the interesting ones. Like, what am I going to do? The gene that makes your hair color? None of you are bald, so it's not like that's like a big deal to talk about.
Daniel's kind of bald.
Yeah, Daniel's a little bit bald.
Yeah.
On the back right of his head, if you want to turn around and show everybody.
You could probably not have pointed that out, and he probably would have been fine with it, I think. Yeah. You've never eaten beets before either, have you? Do you eat vegetables?
I eat carrots and tomatoes.
Those don't do anything interesting.
I think, don't carrots.
Oh, but carrots, if you eat too many, you turn orange.
Yeah, my cousin.
I don't want to.
Yeah, but that's like a lot of carrots. It's like gets you like.
You gotta be a rabbit. Yep.
Maybe like your sister chromatids, are those together?
Yep, they're attached at the centromere.
Oh my gosh, I didn't know that was a thing.
This becomes important to us because this is going to be a major player in mitosis. So yesterday, we looked at this particular process, the cell cycle. And I told you what did I do wrong? Like a delinquent diaphragm here. I told you yesterday that it's a cell cycle. It occurs as a circle, right? The beginning is the end, and the end is the beginning, yada, yada, blah, blah, blah. If you interpret this chart as a pie chart as well, so if you look at it like so, this is 100%. This section is interphase. And this section is mitosis and cytokinesis. If you look at it and you're like, yes, it looks like 85% of it is interphase. So the cell spends 85% of its life in interphase and 15% of its life in mitosis. That would be accurate. Whoever taught you how to interpret a picture taught you well. Thank you. 6th grade math teacher? Did you like your 6th grade math teacher?
No.
Okay. Thank you, 5th grade math teacher.
Yeah. I like my same teacher.
Thank you, 4th grade math teacher. There's like 2 of you who are like, yeah, okay, fine.
8th grade.
Thank you, 5th grade science teacher. Did any of you like any teacher you've ever had? Man, I should have gone into a job where people might actually maybe like me. Good grief. You guys are a hard sell. Puppies, kittens, butterflies, unicorns. This, you'll notice that this section is the only section we haven't talked about, mitosis. Hasta luego. Oh, I can say good luck. It is not one of those events in which it's bad luck to say good luck. Good luck.
Thank you.
Play well. Avoid flying balls, unless your job is to catch them, in which case, catch them.
Bye.
I'm going to give you 4 minutes to copy these little pictures down. It's going to be important that they're in your notebooks. Some of you are going to try and take a picture with your iPads. I'm going to remind you what I have reminded you of in the past, how your brain synthesizes information. It's most productive when you do so with your hands. And friends, if it's like super tiny and you're one of those people making this face, you may come up and take a look at it. I don't mind. I don't have that many personal space issues.
Take a picture of it and then
This whole process that you're looking at here is called mitosis. So this is what happens in that small 15% of the wedge. I should point out to you that very first cell that we drew on the very first slide, that one is what a cell looks like in interphase. So in the other 85% of that wedge, or of that pie chart or circle or whatever you want to call it. That means that you see a nucleus, you see a nuclear envelope. You can't see chromosomes because they only condense and are only visible during mitosis. So you will only ever see chromosomes during mitosis. The first stage of mitosis is prophase. And that's the one that you see right there on the left. One of the things that's the hallmark of prophase is that chromatin condenses into chromosomes. Chromatin condenses into chromosomes. You'll notice there's no longer a nuclear envelope. No nuclear envelope at all. It's dissolved. Falls apart. So there's no distinct nucleus any longer. The centrioles begin to migrate to opposite sides of the cell. We do call opposite sides of animal cells poles, just like we would if we were looking at a globe. If it irritates you that the poles are on left and right sides, like east and west, rather than north and south, turn your head. Magically, it becomes north and south. Voila. So it's going to migrate to opposite poles. And those little black lines at the side called spindle fibers are going to form. So spindle fibers form during prophase.
Wait, the things that hold the chromosomes?
Yes, they attach to chromosomes. So what's actually gonna happen here is spindle fibers are going to come out of the centrioles and attach to the centromeres of each chromosome. Consider it something like a fishing line and fishing reel. The centriole is like the reel, and the spindle fiber is like the line. If you're fishing properly, when you reel in the line, there should be a fish at the end of it, if you're any good at what you're doing, or lucky. In this instance, rather than finding a fish at the end of the line, you're going to find a chromosome instead. So at one side of a spindle fiber, you have a centriole, and at the other side, you have a chromosome.
What's that thing in the middle?
Of the chromosome?
Yes.
So that little dot right there?
No, on the first one.
This one?
Yeah.
That's all chromatin.
So all of those happened at once but they made it similar.
So it all happens in the same period of time. Technically, the second picture that you see here labeled prometaphase, most textbooks just call it part of prophase. So most textbooks call prometaphase part of prophase. You'll notice that the chromosomes are completely visible as those little X's now, rather than being like all unfurled as chromatin. And the centrioles have migrated to completely the opposite sides of the cells. Sure. Chromatin is completely condensed into chromosomes. And centrioles have finished migrating to opposite sides of the cell. What's going to happen next is kind of like a game of tug of war between the two centrioles at opposite poles. In a game of tug of war, if both parties are pulling equally, where is the small ribbon tied at the center of the rope going to be? Both teams are pulling equally. In the middle, right? So what's going to happen in metaphase is a game of tug of war between centrioles where they're going to pull equally. Now, if you guys didn't notice, during Spirit Week, I did not participate in faculty-student tug of war, mostly because it seems like a lot of work. Yeah, I don't really feel like doing that. And because I don't really particularly care for following rules. I would engage in some schadenfreude myself and would find it infinitely more entertaining to go cut the middle of the rope. What happens if you do that?
The other teams are just done. Like, they go flying.
They would go flying to opposite sides, right? Same thing happens here. During metaphase, there's that game of tug of war that aligns chromosomes perfectly down the center. The center, by the way, is called, wait for it, this is super creative, the equator. Sometimes also called the metaphase plate. So if I were to ask you where do chromosomes align during metaphase, and you were to say the metaphase plate, that would be entirely and completely correct. Keeping in mind there's not actually a dashed line at the metaphase plate, it's more of a figurative region than it is an actual place. Anaphase is what happens if I were to go and cut those two sister chromatids in half. The sister chromatids are going to migrate to opposite poles. At telophase, two new nuclear envelopes are going to form, and we're going to have two new nuclei. My telophase diagram is this indentation right there. That indentation only occurs that way in animal cells. It's called a furrow, or a cleavage furrow, because to cleave means to cut. So it cuts the cell into two new cells by making indentations. Those indentations are going to meet in the middle as they get closer and closer and closer and closer and end up making two new cells. That is not part of telophase. And it occurs at the same time as telophase, but is not telophase. It's part of cytokinesis. Cytokinesis meaning cell dividing.